The overall goal of the following experiment is to express a human protein in different heterologous systems. For production of a transient protein expression system, bacterial cells that have been transformed to express the recombinant protein of interest are injected into Nicotiana benthamiana leaves, facilitating the accumulation of the protein within the infiltrated leaves only. For production of a stable protein expression system, Nicotiana tabacums plants are transformed by the Agrobacterium leaf disc infection method to obtain transgenic tobacco plants that stably express the protein of interest in the whole plant. Ultimately, the plant-based recombinant protein expression can be evaluated by Western blot analysis.
The main advantage of plant-based techniques over existing methods like bacteria and insect cells is their versatility, combining speed of expression and scalability.
We first had the idea for this method when we realized that the expression of the recombinant protein in bacterial cells resulted in the production of a non-functional protein.
For agroinfection of Nicotiana benthamiana, first inoculate the bacteria of interest in 30 milliliters of lysogeny broth medium supplemented with antibiotics at 28 degrees Celsius. After 2 days, collect the bacteria by centrifugation and resuspend the pellet in 10 milliliters of infiltration buffer. Then measure the OD600 value and adjust it to 0.9 by dilution with the same buffer. Next,